Okay, next up we have Rick Boonen from the Leiden University Medical Centre, and he's going to talk about high-throughput analysis of Parp2 missense variants linking functional impact to breast cancer risk.
Right, thank you for the introduction, and thank you for the opportunity to have us present our work here. So as introduced, I will talk about the high-throughput functional analysis of PALB2 variants, and in the last slide I'll also try to associate these results with breast cancer risk. Before I start, I actually do want to sort of start with a side note that the high-throughput assays that I'm going to show today, although we performed, I think, most of the assays that we wanted to, with regards to the analysis of the data, it's all still quite preliminary, so we still have to, let's say, filter out noisy data and et cetera, and statistical analysis. All right, so we started working on PALB2 a couple of years ago. That's because it's really becoming recognized as a high-risk gene for breast cancer. And you also see this in this graph where pathogenic variants in this gene associate with a similar lifetime risk for breast cancer as pathogenic variants in BRCA2, so that's almost comparable. So that graph is really for variants that are known to impair protein function, such as nonsense variants or... Yeah, of course. Sorry. So that's really for nonsense variants or variants that are basically frame shifting, etc. But as has been discussed quite often at this meeting, it can also be the case that a FUS is found in a carrier. And most FUSs are actually missense variants. So in order to interpret a missense FUS, having functional data can really help to tip the scale either way in classifying a variant as pathogenic or as benign. So if we want to study the function of a missense variant in PALB2, then we first need to look at the function of PALB2. This has also been introduced shortly yesterday, but PALB2 together with BRCA1 and BRCA2 actually has a crucial role in this pathway called homologous recombination, which is a repair pathway that mediates the error-free repair of a double-stranded break in the DNA, so thereby maintaining genomic stability. So PALB2 actually functions together with BRCA1 and BRCA2 in this pathway. And because they have such a crucial role there, we can use that pathway as a readout for protein function. So this is something that we can do here in our mouse embryonic stem cell-based system. So our cells are knockout for PALB2. PALB2 is an essential gene, so we first had to knock out P53, because then basically these cells tolerate the subsequent knockout of PALB2. Then looking at the ROSA26 locus at the middle, we have integrated these recombination sites that allow us to fairly efficiently flip in our human cDNA for PALB2. And after that stable integration, these cells then also become resistant to neomycin. So then looking at the top, at the PIM1 locus, we have integrated the Drgfp reporter. So this is our reporter to measure homologous recombination. The way that works is as follows. So if you transiently express this IC1 endonuclease, then it will make a break, a cut in the non-functional GFP sequence, so the SCE GFP. If that break is repaired through homologous recombination using the template that is located further in the reporter, so the IGFP, then that will result in an intact GFP gene, expression of GFP. So this is then something you can measure with FUX analysis. So the amount of green cells that you see is a measure of the HR efficiency. So then as a second functional readout, we can also use sensitivity to the Parp inhibitor. So if we treat cells with a Parp inhibitor, then Parp1 will be trapped near sites of single-stranded DNA breaks. And that trapping there can result in the formation of a double-stranded break at those sites during replication. So cells that, for instance, express a functional variant of PALB2, so cells that are proficient in HR, are fairly resistant to that, to the Parp inhibitor, whereas cells that, for instance, express a damaging variant of PALB2, they cannot repair these breaks in an error-free manner, and they are sensitive to this type of inhibitor. So these are actually two assays that we can use to look at the function of PALB2 in homologous recombination. So then the question is, which PALB2 missense buses do we actually want to test? So then looking at ClinVar, we see, maybe the number has changed by now, but we see that roughly 1,985 PALB2 missense buses have been reported. As you can see, they're scattered quite evenly over the exons of PALB2 and over its functional domains. But because there are so many, so yeah, then the question comes, how do we address this large number of variants, and where do we actually start? So in our previous study, we and others have already found that damaging missense variants appear to localize to the WD40 as well as the coiled coil domain of PALB2. So looking at the blue boxes at the bottom, those are all of the missense variants, and indicated in red are the damaging ones. And they really localize to the coiled coil and WD40 regions. So the coiled coil region is actually important for the interaction with BRCA1, whereas the WD40 region is important for the interaction with other HR factors such as BRCA2. Now, we have not identified any damaging missense variants in the middle region of PALB2 yet. And actually consistent with that, we see that when we take out the entire CAM domain, or the entire MRG15 domain, or the entire exon 4, which is the largest exon of PALB2, we see that has no effect on PALB2's function in homologous recombination. So these deletion variants were all measured with the Drgfp reporter. So this basically indicates that these regions are not critical for PALB2's function in homologous recombination. And we reason based on that the identification of damaging missense variants in those regions will probably be unlikely. Although it's not direct evidence for that. So hopefully we can compare this statement to the data from Leah Starita's lab quite soon. So yeah, with the high throughput analysis, we really wanted to be pragmatic and focus only on the coiled coil and WD40 region. So we actually started with the coiled coil region because it is quite small. We obtained a variant library for that domain through collaboration with a company. And in the next slide, you can see the diversity of all of the protein variants in that library. So the coiled coil region goes from leucine number 9 to lysine 43. So that's 35 amino acids. So for that region, you then have 700 variant possibilities. So 665 missense variants and 35 nonsense variants. Now, roughly 96% of all of the variants were detected in that library. And as you can see, the variants are present in slightly unequal proportions, I would say. So it is a bit harder to get reliable data for variants that are not so abundant in the library. So then going to the setup of our high throughput functional assay. So we have our PALB2 knockout cells. We can stably integrate the variant library. Every cell will express a different variant. And on that pool of cells, we can perform a Parp inhibitor treatment assay. So we then take a population that is not treated with the Parp inhibitor and compare it to a population that is treated with the Parp inhibitor. So in the treated population, you then expect to see a depletion of all of the damaging PALB2 variants. So from those populations, we can isolate DNA and subject it to next generation sequencing and variant calling by Enrich2. So then looking at the results for this coiled coil region, it looks as follows. So in the bottom row, you see all of the nonsense variants, and they are all nicely depleted after Parp inhibitor treatment. So that's basically a first validation that our assay is working quite nicely. We furthermore see that damaging missense variants localize to several leucines, so 21, 24, 35, but also tyrosine 28. And we see that a change into a proline is quite often damaging at multiple sites, which is also not surprising because I think it can really disrupt the structure of a protein, at least locally. But most importantly, these results actually address almost all of the missense buses that have been listed in ClinVar for this region. Now, 5 of those are actually just as damaging as the nonsense variants. So hopefully these results will contribute in the end to the reclassification of such variants to likely pathogenic. So these results were validated in 2 more ways. So based on the high throughput results, we also selected 33 variants that we then tested in single Drgfp assays, which is displayed on the x-axis in this plot. So then we see actually that the measured HR efficiency correlates with the depletion score, so the high throughput score. And as a third validation, from those 33, we selected 8 more to test the interaction with BRCA1. We used the YFP PALB2 construct for that, so being able to pull down the YFP tag. And then we nicely see that when we pull down a functional variant for PALB2, so the 2 green ones actually, as well as wild-type PALB2, then you see in the top plot of the bottom 3 that you nicely also pull down BRCA1 as well. Whereas for all of the variants that showed impaired function, we see that the interaction with BRCA1 is also impaired. And actually the yellow ones were intermediate in our high throughput assay, and for those you still see a subtle interaction with BRCA1. So this is all really nicely consistent, I would say, and it shows that we can use our mouse embryonic stem cell based system for this type of high throughput assays. So a question that I kept on getting back home was, so do different nucleotide variants coding for the same missense change actually give the same result? So I simply picked the 7 most abundant missense variants in the library because I guess for those you expect the most reliable results. For instance, missense variant 1 has 2 codon options in the library, number 2 also 2, etc. And I think the results do compare quite well between these different nucleotide variants leading to the same missense variant. So that's sort of what you would expect. Yeah, sort of an observation that we also made. It's not a very strong effect, but we see that functional PALB2 localizes mostly to the nucleus. But when we have an intermediate or a damaging variant, it tends to localize a little bit more to the cytoplasm. We cannot really discriminate between intermediate and damaging variants with this assay, and it's quite a mild effect, I would say, but we do see it quite consistently. It doesn't really compare to a damaging and unstable variant, which is the last condition. So that variant, I think, probably because it is extremely unstable, it localizes mostly to the cytoplasm. All right, so then that actually brings me to the second part, which is the WD40 domain. So this region is actually 10 times larger than the coiled coil region. So for this, we wanted to keep the experiments quite similar to the coiled coil region. So we split this region in 5 different parts, and we got a variant library for each part. So these libraries are coming from Twist. So here we only have 1 codon option coding for a missense change. So it's different than the coiled coil library. And you also nicely see that all of the variants are present in fairly equal proportions. So we have high throughput results now for the first 4 libraries. The 5th one is actually still ongoing. So those results we can look at again in the form of this heat map. So everything is a bit smaller now because the map is bigger. But we again see in the bottom row that all of the or at least almost all of the nonsense variants are depleted after PARP inhibitor treatment. So again, sort of validating our assay. The results also correlate with Drgfp assays for some variants that we analyzed already and for which we published the results already. But then at the bottom, most importantly, these results address more than 400 missense fusses that are listed in ClinVar. Now, roughly 40 of those are damaging, I think. I only counted the ones for the first library. There were 13 there, so it's sort of times 4, let's say. And what you also see from this heat map is that that several sites are really crucial for PALB2's function in HR. And I actually expect that most of the variants there that are damaging are critical for maintaining the stability of the PALB2 protein. And that's because we identified a couple of variants in the WD40 domain already, and for those the results are also at least in part published, but all of them actually impaired protein stability. And we confirmed that with this cyclohexamide treatment assay for 2 variants. So cyclohexamide actually stops the synthesis of new proteins, so you can look at the pool of PALB2 protein over time. And for wild type PALB2, going from 0 to 3 hours, you see that the pool of PALB2 protein remains quite stable over time. So and then for the 2 variants, you see that pool of protein actually goes down. So you need to look at the bottom band, by the way. So the top band is actually a background band. But then what we actually hope to find still are there missense variants in the WD40 domain that are damaging but still stable. So variants that are hopefully affecting an interaction with, for instance, BRCA2. So hopefully we can pick some variants based on the high throughput results and find some of those. But so far we haven't. So then to sum up so far, going back to this introductory slide, so with the coiled experiments, we addressed almost all of the variants in ClinVar. Based on the deletion variants, you can reason that these variants are less likely to be damaging. Of course, it's not definite proof for that. But then for the WD40 region, when we're done at least, we hope to have addressed almost all of the variants in ClinVar as well. And then by the end of this, we may have addressed roughly 85% of the variants listed there. So in the last slide, I just want to try and associate our functional results also with breast cancer risk, which we can do on the basis of this study from the Breast Cancer Association Consortium that was published last year. So they sequenced a large number of cases and controls, and they identified more than 500 distinct PALB2 missense variants. Now, for 89 of those, we actually performed Drgfp assays. And in the graph, you see 2 thresholds. So the green one is actually based on several likely benign missense variants that we tested. And the pathogenic threshold is based on 12 truncating variants that we tested. So in this graph, you can sort of group all of these variants based on function. So when you look at the top group, you can associate that with cancer risk, so with an odds ratio. And it doesn't associate with a higher risk for breast cancer. It becomes a little bit higher for the middle group, the intermediate group, but definitely not significantly. But then when you look at the bottom group, so the small group of missense variants below the red line, it's still a very, very small group, but they, as a group, associate with an odds ratio of around 6, which is considered a high risk for breast cancer. And it actually compares quite well to what has been reported for nonsense variants or truncating variants in PALB2. So overall, I think that really highlights the importance of identifying damaging missense variants. And hopefully I can quite soon sort of remake this graph based on the high throughput data, but that is still ongoing. So hopefully these calculations will also become better, because the bottom calculation is, you see that it has a very wide confidence interval, and it's still based on very low case control frequencies. All right, so with that, I want to thank everyone in Heiko's lab, and Heiko, of course, for the supervision on this project, and especially Sabine actually performed a lot of the assays that I showed today. If there are any questions.
I'm really curious. So you have all these WD40 domains. Have you tried aligning those different mutational landscapes to find residues that are conserved across WD40 domains that are necessary for them?
I haven't looked at that yet. So I think generally the WD40 domain is quite well conserved, but I haven't looked at whether these let's say these hotspots that you see in the heat map are more conserved than other regions. So I still need to do that actually. Yeah.
So I have a question. I was wondering if you had any insight into the mechanism that causes aberrant mislocalization between the nucleus of the cytoplasm.
No, actually, no, I don't. So it seems like the interaction with BRCA1 is required for the correct localization of PALB2 in the nucleus. But so maybe BRCA1 sort of keeps PALB2 in the nucleus, and if it can't interact, it moves out. But I definitely don't know how it works. Yeah.
Hey, I'm curious about your validation assay, the Drgfp.
Yeah.
Would it be possible or of interest to use it as a full-scale deep mutational scan, not just as a validation, if you combine it with like flow cell sorting?
Yeah, so we were hoping to do that. So the problem with that is that when you perform a Drgfp assay, let's say for a functional variant, with the mouse cells you get roughly 10% of green cells that are positive for GFP. And I tried it in the beginning with the coiled coil library, but it took a long time to sort enough green cells. So in the end, we didn't try to optimize that, and we chose for a more pragmatic approach, which is the Parp inhibitor assay. But it should be possible. I think Leah has done it. But we chose a different tactic. Yeah.
Okay, that makes sense. So if, for example, like a counter-selectable marker was included, maybe you could increase that 10% and actually get a good proportion of cells sorted.
Sorry.
So if you use like a counter-selectable marker for the validation assay, so that if the homologous recombination is successful, you also have expression of a marker that you can then select for. In addition to
but you can select on GFP, right? That is your marker, I guess.
An additional marker just to increase efficiency. But yeah, I see the challenges, and the Parp inhibitor assay looked really good. Thanks.
Thank you.